Welcome back for another session of RNA biology, and this is the last slide where we left in the previous class, and we were discussing about the problems with the DNA. Why DNA is not a likely candidate for the storing of the genetic information, etc. So we have seen that while DNA can be a very good prospect, however, it comes with certain limitation, and one of the major limitation of the DNA that it is less reactive. It is very stable and less reactive. So we should understand, in order to perform some task, the molecule need to undergo some activity which in turn is antagonized by the stable structure. So you don't want an unstable structure. However, the extreme stability of the DNA renders it reasonably inactive when a catalysis is concerned. So that is what we discussed in the previous class. And there are no reports or no experimental evidence was demonstrated to show that the DNA can act as a catalyst even in a laboratory condition. So now let us see what are the circumstances in which RNA got converted into DNA as a storing of genetic or storage house of genetic information. So now, based on the exclusion of DNA as a possible early molecule to start the life on Earth, we should shift back into the RNA as an early suspect for life. And let us see what are the plus point or the benefits RNA has. And it is a very possible molecule to have started the initial reactions on the prebiotic world. And we should try to understand a little bit more in depth that what is actually RNA means. To know that more, to do further analysis on this, we must compare DNA and RNA. And both of them have got a sugar phosphate backbone that is formed by the phosphodiester linkage, because these nucleotides are held together by the phosphodiester backbone. And there are two main differences are there. And what are those differences? One is the pyrimidine thymine is preferred in the DNA. It do not exist in the RNA. Instead, the RNA contains a closely related pyrimidine called uracil. And the sugar that is present in the RNA is normal sugar. That means the 2' carbon has got a hydroxyl group. Whereas in the DNA, it is called deoxyribose. That means the oxygen is missing from the 2' carbon. So these are all the two main difference between the DNA and the RNA. So this is a cartoon of the RNA and the DNA. In the left hand side, you have got the RNA molecule. On the right hand side, you have got the DNA molecule. They look more or less the same except for the variation in the thymine versus uracil, and the backbone has, or the ribose sugar has got deoxyribose in the DNA and ribose conformation in the RNA. Other than that, they look more or less the same, and the DNA has got two strands, which also adds to the stability of the DNA molecule. And another important critical point when comparing the two while understanding the DNA is the 2' carbon, which we have been discussing about in the ribose sugar, is much more reactive. That means the 2' carbon in the DNA has got deoxy. That means the OH is now changed into just H. That means oxygen is missing, which makes it less reactive. Whereas in the RNA, the hydroxyl group of the 2' carbon is highly reactive. And this is the main difference that makes the DNA stable. That means the absence of this oxygen makes the DNA more stable. If it is less reactive, then it allows the molecule to be more stable. that is DNA is more stable, the credit goes to the deoxy 2' carbon in the ribose sugar. The opposite is seen in the RNA. The presence of hydroxyl group in the 2' carbon makes the RNA more reactive. And the absence of thymine and the presence of uracil makes them easy to distinguish when various enzymes like various RNA and DNA modifying enzymes make use of this uracil versus thymine to recognize whether a given nucleic acid is RNA or DNA. And now the most important part is the RNA hairpin. We'll see them more in detail. What does it mean? A major difference between RNA and DNA is the ability to form secondary structure. DNA is a very stable molecule, but its secondary structure is limited to the double helical conformation. Both strands are in anti-parallel conformation, whereas RNA has got innumerable conformation possibilities, and we generally call them as secondary structure. Very often, the RNA is denoted as a single strand for convenience. Ideally, it is a single strand, but does it stay like a single strand? The answer is no. It can allow a lot of variations, and also it can undergo pairing similar to that what you see in the DNA. That means some places it can even form helical conformation, and it can form secondary, tertiary and even quaternary conformation, which makes it a proper three-dimensional structure, which is a necessity for carrying out biological reactions. When RNA can appear to be a double-stranded even while staying as a single-stranded molecule, and we usually call this double-stranded structure as a hairpin structure. And this is happening when the secondary structure of the RNA loops forms a double-stranded stem. We can see in a pictorial manner in the next slide. What you see here is a loop region. That means it do not form any pairing, it do not find any complementarity. Whereas in the bottom, as you come down, you see some pairing. That means Watson-Crick base pairing. Like we told you in the yesterday's class, A pairs with T in the DNA, or A pairs with U in the RNA, and G pairs with C. So A is adenosine, U is uracil, G is guanosine, and C is cytosine. And in the bottom, what you are seeing, the so-called stem structure is similar to that of the double helical structure. Such a molecule we call it as hairpin structure, or it's also called as RNA hairpin. And the RNA hairpin stabilizes the molecule and also allows the molecule to undergo different conformation. And RNA can also contain information. We will see what kind of information they are. RNA contains sequence of bases that is analogous to letters in a word. We know if someone writes a English letter R, we call it as R, symbol R, letter R. But also there is a word A-R-E, R, that is also pronounced as R, but both have got a different meaning. Same way, a letter, number of letters present in one word decides how that word is pronounced, and also what is the qualitative meaning that word is imparting. So like that, the sequence present in the RNA can contain information that can specifically mean something. It means something, and we'll see them what does that meaning. And this also allow to carry the information, because the hydrogen bonding that occurs specifically between AU and GC. AU is double bond, GC is triple bond, and it is always theoretically possible that it can make a copy of itself, because you think about A can pair with U, G can pair with C. So a stretch that has got A, C, C, U, G, C, C, something like that can make a complementary copy. That means it can attract another nucleotide and pair it, and it can make a copy of itself. That will be a mirror image. Like if you seeing yourself in the mirror is not what you actually look like. It's actually you see is a mirror image. And if you reverse it with another mirror, that is your actual image. So let us think about the template and the complementarity. And they always will be complementary to each other. When a strand is there, an RNA strand is there, it will always attract a complementary strand, which is the basis of formation of a stem structure. And when there is no complementarity there, it will remain like a loop structure. When considering the replication process, whenever an RNA or DNA, whatever it is making a copy of itself, it is important to know that this kind of terminology means that there is always a pairing, a region that can pair, or a sequence that is newly made while pairing onto a template. Like template basically means a mold. You may have seen how people make different structures. Like if you have a molten plastic, you can pour onto a mold, and after once the plastic solidifies, it becomes shape of that particular mold. So same concept applies while making a new copy. You need to have a template, and the newly formed molecule will be complementary to that. We call the master copy as a template strand, that is the original strand, and the newly formed, the new strand is called a complementary strand. So the new strand being created is very much similar to that of the template strand, but it is not identical. Now you make another strand from this complementary strand that will look exactly like the template strand. So what you should understand, a complementary strand is a mirror image of the template strand, and you make another complementary strand from this complementary strand, it will be the template strand. So it is called so because it needs to match the template as a perfect complement, complementing to each other. It's like you can, if you look your left hand and the right hand, left palm and right palm, you can join them together. They look like a mirror image of each other. But your right hand and another person's right hand, they look same, but they are not mirror images. So that is the similarity you should know between the template and the complementary strand. So now let us think how we proceed with formation of RNA in the prebiotic world. And we know that it has to happen through the polymerization of the nucleotides. So nucleoside triphosphate plus a catalyst. We saw the borate can provide selectivity. One such abundant mineral that is known to contribute for this catalysis is called montmorillonite clay, which is mineral rich and that can act like the substratum act like a venue for this polymerization to happen. So the initial polymerization could have been carried out by montmorillonite clay that was abundant in the prebiotic world. So if you look into the RNA replication part, RNA dependent RNA polymerase is there in this modern world. Like if you think about any influenza virus or COVID virus or any RNA virus you can think of, they always depend on a enzyme, protein enzyme. Remember, that is called RNA dependent RNA polymerase. We do not produce an RNA dependent RNA polymerase in our body. Humans do not produce. It is a feature of viruses normally because they carry that information in their genome and they make use of the machinery from the host and produce this protein and make a copy of themselves. When you get a viral infection, say COVID infection or influenza infection, like influenza means common cold, you get sore throat, etc. So another example you can see that is known to help in the replication of the RNA that is formed in the prebiotic world is the ribozymes, which should work on a large number of sequences. Remember, in prebiotic world, we are not talking or considering about enzymes made of amino acid or protein enzymes are missing them. We do not have a protein enzymes. So the ribozymes have to take the job of making a copy of itself. So coming back to the myth, a small RNA that arises de novo and can replicate efficiently and in high fidelity under plausible prebiotic condition. Why it is referred to as myth? Because nobody was there to witness it, and this is too good to be true. But if this truth hasn't happened, there is no way you could have had any life form existed, because it all originated from the RNA, without which it is out of question. The spontaneous formation of life is out of question. Now let us think about a little bit of energy, addition of energy. How does that contribute? Because many biological reaction, no matter whether you talk about ribozyme or whether you talk about formation of RNA itself, independent of ribozyme, everything requires energy. In order for the template strand to make a new complementary strand, an input of energy is must, and it can come from various sources. It can even come from the UV radiation. And compared to a protein, RNA is very stable and is not very catalytically reactive. Compared to DNA, RNA is very catalytically reactive and biologically active. But compared to a protein, it is less active. But the life forms have to depend on this less active RNA in the prebiotic world. But the times available was so high that this RNA, the so-called ribozymes, even with its slow activity, it was able to perform the task of making a copy of itself. Remember, the early ribozymes or the early RNA molecule that had a catalytic role was always bothered about making a copy of itself, because its goal is to survive. However, in a very rare occurrences, RNA can also form necessary tertiary structures and transition forms that can make use of chemical energy, that is chemical catalysis can occur, and there are evidence even in modern world to show the ribozymes can perform the chemical catalysis of various nucleic acids. So let us see how good or how bad an RNA can be as a catalyst. If RNA can form necessary tertiary structure, necessary is a loaded word, which means that it should form the structure which actually is going to be useful, and there is no director sitting or there is no guide sitting to tell that okay, this is how you should be folding so that you can perform this task. So we should understand the folding is happening random, and the functioning, the output also is random. Now the question is, are this random folding and the random function are going to be beneficial for the survival of this molecule? So they have to have a necessary tertiary structure to behave like a modern day catalytic protein, and then it should be able to break the bonds to release energy, because to perform a task you need energy. Either it should be a physical energy or it should be a chemical energy. If the bonds break, then the free energy is released and the energy could be absorbed by a templating molecule, because then a phosphodiester bond can be easily formed and a copy of itself can be made. So this molecule would have the necessary energy to form the necessary bonds and of a complementing structure, and this could effectively end up in replicating itself. So what are the points we covered so far? We should have a clear template, and that template should have necessary secondary structure which is powerful enough to capitalize the chemical energy from whatsoever possible surrounding, and it should be able to catalyze the polymerization of nucleotides which is complementary to the template, so that you end up getting a new RNA molecule which is complementary to the template. Then it is a successful ribozyme. Now 4 billion years ago, what are we seeing? We had abundant UV radiation. Remember, we don't have too much of oxygen and hence not too much of ozone, and hence there is no protection from UV radiation. And the atmosphere was quite porous for the UV radiation, and plenty of UV radiation was abundantly available during that, which acted like a physical source of energy in early atmosphere. Plenty of carbon dioxide, plenty of nitrogen, carbon monoxide, hydrogen sulfide, methane gas, and oxygen was nil, because oxygen was produced much later in the excuse me. Now let us think about in early ocean, what are the points that contributed to the production of ions? Various ions that were present in the ocean water also could have contributed to maintaining the proper pH for a biological reaction, and the hydrothermal vents also allowed the temperature to be kept quite high. Now, coming back to the RNA world, although we can say in a generic form of natural selection, only certain types of macromolecules that resisted degradation. There is a saying, right? Like in a war field, if you kill or you be killed, that means the molecules also have, like you can see in if you go to any national park or wildlife sanctuary, there is no rules and regulations, etc. So if a lion or a leopard is hungry, it will just go and chase a buffalo or an animal and it will eat. And if buffalo want to leave, it should flee or give a fight. It may or may not win. So it is free for all. Lion is hungry, it will attack. Leopard is hungry, it will attack and it will eat. Same logic applies at the molecular level also. So those macromolecules resisted degradation. It is available for doing something or just be there. It can do something at a later time point. So it has to protect itself from the entropy that could exist longer than any other. So you think about it. If 10 molecules are born, one molecule can live only 10 minutes, then it disappears. Whereas the other molecule can live for 10 hours, it will stay for long. Another molecule can live for 10 years, that means it will be much represented. So this is nothing but stability of a molecule, allow or give the opportunity for this molecule to perform some possible task in the future time. Over the time, the soup became dominated by those macromolecules that were resistant to the degradation. This domination would be brief in large time scheme because this molecule lacked the ability to template replication. Remember, if a molecule cannot do any next step, then it will be dominated and it will be stuck there. You may have heard about Liebig's law of minimum in some of your biochemistry class, which basically means, say, for a reaction to happen, if 10 molecules are needed, okay, 9 of them are super abundant, but 10th one is least abundant. Then the rate of reaction is dependent by the least available molecule. So that decides whether or not the reaction has to happen. In the same way, here the ability of a dominant macromolecule to proceed further decides whether or not that molecule is present in this current environment. If it is present, that simply indicates it is stable. Of course, it is available, but it do not have a downstream track. And now let us see how RNA acts as a dominant molecule. At some time point, RNA molecule, through random chance on and bonding, the hairpin loops forming ribozymes can eventually perform a catalytic role, and we call it as RNA catalyst. And this is a random event. Now you imagine if a given RNA molecule, by virtue of its sequence, which in turn allows a proper secondary structure to form, it now can easily make a copy of itself. Then what will happen? All these macromolecules that are present can technically get converted into those RNA. Here macromolecule we are referring to the intermediates of RNA. So this molecule can then split through catalysis and then form a template with free floating nucleotides. So then if there are 10 molecules that have, say, an RNA molecule that has got 10 bases, another RNA molecule that has got 50 bases, another RNA molecule that has got 200 bases, none of them are able to make a copy of itself. Now comes a new RNA molecule that is having 200 or 500 base, which can make a copy of itself. Then what it will do? It can catch hold of these non-replicating RNA molecule and cleave its bond and use that energy and try to make a copy of this newly formed molecule. As a result of which you can end up having a dominant soup of molecules that have this ribozyme or this RNA that has got a catalytic role. So this dominance is allowing the propagation of this group of RNA molecules. So this allows it to rapidly replicate itself, giving it a chance to increase its number against the tide of entropy. Remember, entropy is always there. The molecule is always challenged. So it should be able to resist itself from the degradation. Over time, this replicating RNA became dominant in the prebiotic soup. So I will end the class with this and we will resume in the next class.